Good morning. For this week's pre-lab video, or this portion of the pre-lab video, we're going to go over the tests that we did from the previous week. You inoculated these cultures, so you'll pick them up this morning. We did, as you recall, a variety of tests. We inoculated methyl red, Vokes Proskauer media, MRVP, a tryptone broth for a test called the indole test. We did citrate, agar slants, and we are testing for the decarboxylation of two different amino acids. So in some of these tests there are excuse me, reagents that need to be added, and in one of these particular tests the incubation time for looking for color development is about 15 minutes. So just to make the most efficient use of your time, we're going to do that test first, and hopefully by the time this video is finished we can see the results. But if not, you have to do this incubation for the Vokes Proskauer test for about 15 minutes. So you should find these little transfer pipettes at your bench, and what you're going to do is take your MRVP broth. You've inoculated various different species. We're not going to worry about sterile technique at this point because we're going to autoclave all of these bacteria afterwards. But I just wanted to show you, you can tell that the bacteria have grown because this is an uninoculated, good morning, this is an uninoculated tube on the left. This is a 48-hour culture of an enteric bacterium on the right. So to do the Vokes Proskauer test, I'm going to sterilely, although sterility is not important here, take about 1 ml of that culture. The remaining portion of the culture is going to be used for the methyl red test. I'm going to transfer that roughly 1 ml to another glass tube. And again, sterility is not important, but keep in mind that you have now put a potential pathogen into this Pasteur pipette, so make sure that this gets disposed of properly. Now, to that 1 ml, you need to add 2 different reagents and then wait for 15 minutes. And those reagents are 40% potassium hydroxide, so recognize that that's a strong base. So be certain that you're wearing proper protective gloves and lab coat. And a chemical called alpha naphthol. So we'll start with the potassium hydroxide. You're going to add 0.2 of an ml, which roughly equates to 5 drops of potassium hydroxide. 1, 2, 3, 4, 5. I'll mix that up slightly. Because it's a base, I'm going to make sure that I cap that securely and set it back. And then the alpha naphthol, I'm going to add 0.6 of a ml, so 3 times that volume, or 15 drops. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So you can see what the starting culture is. What we're going to look for is over the next 15 minutes a pink color to develop in that. Positive for the Vokes Proskauer test is a pink color development, and that would indicate the production of butanediol fermentation. The remaining portion, so while that's cooking, to the remaining portion of the MRVP broth, we're going to use the remaining portion of that culture for the methyl red test. So methyl red is nothing more than a pH indicator, and it's an unusual pH indicator in that it starts out red, or it starts out kind of an orangish color. If this media, after 48 hours of incubation, has produced acidic fermentation products, this will actually turn very red. If, on the other hand, the fermentation products are neutral, and I've added about 10 drops of methyl red to that, and I'm going to mix it up, the media remains orange to yellowish color. So this would be a methyl red negative test. If this were acidic, this would have turned bright red. So hopefully what we'll see is after time, after the Voges-Proskauer test develops, that these two tests are complete opposites. When you have a Voges-Proskauer positive organism, that means that that organism is a butane-diol fermenter. It means one of the end products of fermentation is the neutral product, butane-diol. Whereas the methyl red, if that's positive, means that it's a mixed acid fermenter, something like E. coli that's producing formic acid, succinic acid, acetic acid. Hopefully these two tests should be mutually exclusive for one another, but we're going to set them aside because you can't really tell for certain if the Voges-Proskauer test is positive yet or not. The only other test from last week that you need to add a reagent to get a positive or to get a readout is the indole test. And again, what you did was you inoculated a tryptic digest. So this is a very amino acid rich media. This is the sterile media here. You can see that it's very clear. After 48 hours, you can obviously tell that your bacterium has grown to great density. Now what we're going to do is assay for an end product of tryptophan hydrolysis. So if this bacterium breaks down that aromatic amino acid tryptophan, one of the waste products that's excreted into its environment is indole. And we can assay for indole using what's known as a Kovacs reagent, or appropriately enough, the indole reagent. So we're just going to pull up a few drops of this, and you can notice that it's kind of a golden color. And we're going to put just a few drops of this into that media. It doesn't mix very well with the bacterial, with the broth culture. And just mix that a little bit. What we're looking for is a very instantaneous change to a very cherry red color. This is an indole negative organism, which was an unfortunate choice on my part. I should have chosen a positive one. But if the color remains the same, that's a negative test for indole. So that would be an organism that is not capable of breaking down tryptophan rather as a carbon and energy source. Some of the organisms that I've had you inoculate last week will turn, with any luck, a bright cherry red on the top. Very striking reaction, but that is an indole negative reaction. The other tests or the other media that you inoculated last week, we can tell the results of without even adding any reagents. So you did inoculation of several different media onto the Simmons citrate agar. So that's this green agar slant. What you're assaying for in this chemically defined media is the ability of the bacterium to utilize citrate as a sole carbon source. There is no other carbon source in this defined media. So if you get this brilliant royal blue color on here, what's happened is the bacteria has grown, utilized citrate, and there's a pH indicator in here called bromethymol blue, which turns blue under acidic conditions. So even though you can't really see much evidence of growth when you look at this, because this is not a particularly great media for the bacteria to grow on, in fact it has only one carbon source, if it is growing on here, it produces an acidic reaction which turns this media from green to blue. So a very easy reaction to determine whether something is positive or negative. And in terms of determining your bacterial unknowns, this will be an important test. Also, it's still an important test clinically to this day. So that's testing for citrate positive. The other two media that you inoculated last week were amino acid decarboxylase media. So in one case, you inoculated lysine. So this is the way the lysine media looks before inoculation. And if the bacteria grows in this, and all of the bacteria that I gave you to inoculate will grow in this, you'll get either a little bit of turbidity, indicating that bacteria grew, but basically no color change, or possibly even a color change to acid, which is yellow, which would turn it more yellow than this. A positive reaction would be demonstrated here in this ornithine reaction. So this is the way the ornithine media looks prior to inoculation, a light tannish color. What happens in a bacterium that can decarboxylate lysine to a really bad smelling chemical called putrescine, is that carbon dioxide and ammonia are released. And so, especially due to the release of ammonia, you get an alkaline reaction. The pH indicator in here turns yellow at acidic conditions, and it turns purple under alkaline conditions. So what you're seeing here when the purple color develops is a positive lysine decarboxylase organism. What's happened is that by putting a vapor barrier on top of this with the mineral oil, you've allowed the bacterium to utilize the small amount of glucose that we put in that media. So it ferments that glucose and utilizes the oxygen in that media. Because the mineral oil vapor barrier prevents oxygen from coming or from diffusing back into that media, it sets up an anaerobic environment. It turns out that amino acid decarboxylase enzymes are induced only under anaerobic conditions. So this is why it's really critical that you always place the mineral oil vapor barrier on top of the media before you or after you've inoculated it. If you don't do that, then your test is inconclusive. This would be a positive lysine decarboxylase organism, as opposed to this organism, which is negative for lysine decarboxylation. Now, let's see, there hasn't been that much time gone, but if you look now at the Vogts-Proskauer test, and if you look carefully at the top of that, you're beginning to see the development of a red color, a pink color near the top of that. That indicates the presence of one of the intermediates in the fermentation pathway of butanediol called acetyl-methylcarbinol. But in either case, this positive reaction in Vogts-Proskauer indicates a butanediol fermenter. So this is going to be a very important test for you to understand for the lab practical, but also very important for you to be able to perform when you do your laboratory unknowns. And so in the next part of the pre-lab video, we'll go over your inoculations for next week, as well as the oxidase test. Okay, so for the remaining portion of the pre-lab video for next week, I'm not going to bother to have you watch me inoculate urease broth and carbohydrate broths. You know how to do that. So I'm going to leave that to your own devices. But there is one test that you're going to do this week in the laboratory that you'll get the results of immediately, and that's the oxidase test. And we discussed this in lecture. This is an assay for a specific terminal cytochrome oxidase. So this is something that is found predominantly among obligately aerobic bacteria, that is, in other words, bacteria that can only use oxygen as their terminal electron acceptor. There are some exceptions to that. But for the most part, if you get a positive oxidase reaction, that means that that bacterium is incapable of doing respiration with any terminal electron acceptor other than oxygen. So if we take a facultative anaerobe, such as Enterobacter aerogenes, this is an enteric bacterium, a pathogen that is related, or it's in the same family as E. coli, the Enterobacteriaceae. I'm going to take a pretty good slug of that bacteria on a sterile loop, and I'm going to rub that onto just a very small area of just a piece of filter paper. You'll find filter papers cut to approximately, 3 by 2 centimeters or something at the benches. And then I'm going to sterilize that loop before I set it down. Then, to compare that, we're going to use the opportunistic pathogen Pseudomonas aeruginosa. So this bacterium, you can actually see some green color to it. It produces a beautiful green pigment with extended growth. And I'm going to take this bacterium, and I'm going to take a pretty good slug of that bacteria, and I'm going to put it on a separate spot on there. You should be able to actually see the bacteria on the filter paper. This filter paper is dry. It's not pre-wet with anything. And again, always be certain that you sterilize the loop before you set it down. And we don't need the gas anymore, so I'll just shut that off. So now, I mentioned in lecture, and you don't need to know the name of the chemical, it's a phenylene diamine. It's not quite colorless. It's a little bit purple, but I'm just going to add a couple of drops, one to each bacterium on each side. Now the reaction here, if I'm lucky, is close to instantaneous. So I'm beginning to see if I can pick this up. So you couldn't really see much color to the bacteria before I put this on here. But on the right hand side, you're beginning to see the development of a purple color. So within about probably 2 minutes or less, if you don't see a pretty distinctive purple color on the oxidase test, it's a negative. The organism on your left, Enterobacter aerogenes, is a facultative anaerobe, and so it's not turning purple. There is some color there, but it's not a purple color. And the Pseudomonas aeruginosa is turning purple, albeit not as strongly as I would have liked. This is this plate, as you can tell from the fact that it has a lot of pigment in it, is a little bit old. So these cells might be a little bit sleepy. But you'll see with fresher cultures, hopefully a very strong purple color for oxidase positive organisms and no color change for facultatively anaerobic bacteria, oxidase negative tests. For the most part, so for this test, you won't really need to use this in your unknowns, because all of your unknowns, all of your gram-negative unknowns, will all be oxidase negative. They'll all be members of the same family as Enterobacter aerogenes. In fact, some of you will get Enterobacter aerogenes. So there will be no oxidase positives among your unknowns. Other than that, in this week's lab, you will simply be inoculating media such as the urease broth, lactose broth, sorbitol broth, and a broth where we're going to assay for the reduction of nitrate to nitrite and to nitrogen gas. But those are just straight up inoculations that you know how to do, so no reason to take up your time on this video. Okay, I'll see you in lab.